Bismillahirrahmanirrahim. I hope that all of my students are getting well in this pandemic situation and preparing for the upcoming exams at home and boosting up the immunity. However, today I, Rokshanara Pervin, Senior Lecturer, Department of Biology, Milestone College, welcome you all to the online class from Chapter 10, Zoology. Today's topic is, as you expected, is immunology, of course. So today's topics are innate immunity, acquired immunity, and the steps of acquired immunity. So as we know, the innate immunity is known as the non-specific immunity, or another name is genetic immunity. Why this is genetic immunity? Because this immunity occurs naturally as a result of a person's genetic constitution or physiology and does not arise from a previous infection or vaccination. It is a non-specific defense mechanisms that come into play immediately or within hours of an antigen's appearance in the body, which includes different components such as intact skin, salivary enzymes, neutrophils, natural killer cells, and complement proteins and other enzymes too. That means, in short, the innate immunity also include, first, second, and third line immunity too. So this sort of non-specific immunity can be found in all the species, including the human body, from the birth. Okay, so let's discuss in details about the innate immunity. There are three categories of innate immunity: species, racial, and individual. You know that species means species specific. That means the smallpox viruses never can actually infect the cows or cattle, whereas the cowpox virus, which appears to have species immunity, to the closely related to smallpox viruses. In this sense, smallpox is quite deadly in the disease of in case of humans, but the cowpox is mild, which is localized in skin infection. What about the racial immunity? Some northern Europeans appears to be more resistant to tuberculosis than are the most Africans. On the other hand, Africans are naturally resistant to the variety of African diseases that readily kill the whites. So racial immunity is a form of innate immunity which is shared by the most of the members of a genetically related individuals or population. And individual immunity, it doesn't matter actually whether they are living or what kind of species or any kind of relationship. So this immunity is a form of innate immunity which is not shared by most of the other members in some race, which is shared in other race. So it is variable. Okay, what is acquired or specific immunity? Active immunity is one part of specific immunity, and another part is passive immunity. So let's discuss about the active immunity. So acquired immunity or specific immunity is actually acquired from by the attack of the specific agent of a disease or by the injection of antigens, which is called the acquired immunity. It is very specific and prevents further attacks. It may last for whole life or maybe some years. It is divided into, I told you, active and passive. So active has some subcategories like cell-mediated and humoral immunity. So cell-mediated immunity is can be defined into three more categories like cytotoxic T cells, helper T cells, and suppressor T cells, which are mainly T lymphocytes. So they play important roles in killing germs. They are also called the killer T cells or CD8. Plus the cytotoxic cells are the lymphocytes that destroy infected cells, tumor cells, cancer cells, and provide protection to our body. And helper T cells produce from the lymphocyte and releases cytokinins. cytokines can activate the T cell, build B cell, and accelerate the phagocytosis mediated by macrophage and other phagocytes. These cells also contain TCR, which can recognize the specific antigen. However, the suppressor T cells suppress the activities of the previous cells. Let's discuss about the humoral immunity. What is the humoral immunity? Some B lymphocytes produce memory cells, clone which can be activated as plasma cells. These plasma cells stay within the lymph nodes and produces antimicrobial immunoglobulins, which are called antibody. These antibodies are found in tissue fluids and spread throughout the body. These antibodies bind with the pathogenic antigens and neutralize the pathogens. Okay, and humoral immunity has further details, but for your syllabus it's enough, I guess. And the another types of active immunity are naturally acquired immunity and artificially acquired immunity. So naturally acquired passive immunity can be also categorized into two parts: naturally acquired passive immunity and artificially acquired passive immunity. The examples of active immunity are well written in the book, and I have already mentioned in the first class about the details. So the passive immunity. Passive immunity is a type of immunity which is acquired by the transfer of antibody from one individual to another, such as from mother to offspring. Passive immunity is also of two types. One is naturally acquired, which occurs when mother transfers antibodies to her offspring by placental route during pregnancy and via colostrums during breastfeeding. Immunoglobulin G or IgG is the only antibody isotype that can pass through the placenta. What of the artificially acquired passive immunity? Yes, this is a short-term immunization by the injection of antibodies such as gamma globulin that are not produced by the recipient's cells. Passive immunity acquires artificially is done by the injecting antibodies or sensitized lymphocytes to an organism via a vaccination process or injection of any kind of specific chemicals. What are the basics of killing germs? You see the diagram. So this diagram is all about the whole macrophage, which is the modified form of the monocyte. Macrophages kill the germs by phagocytosis process. At first, bacteria enters the macrophage, forms the phagosome, and the lysosome attaches with the phagosome, forming phagolysosome. Here, in internal digestion or intracellular digestion occurs as a waste kind of excreta, as waste products. Undigested wastes are eliminated from the body. Okay, so these are the cellular responses or the explanation of this diagram. Just make a note on this. Well, here are some important issues related to pathogenic reactions or phagocytosis process. You see in the diagram, when a pathogen such as bacterium is encountered by a phagocyte, the phagocyte extends a portion of its plasma membrane, wrapping the membrane around the pathogen until it is enveloped. Once inside the phagocyte, the pathogen becomes enclosed with an intracellular vesicle called phagosome and finally forming phagolysosome. You see here is a cut or incision on the finger. This is the way this might kill your overall system even you are not protected inside. So it's just a paper cut, but the break of your skin could provide an easy way for the pathogens to enter into the body. If bacteria were to enter through the cut and infect the wound, your innate immune system would quickly respond with a dazing area of the general defenses. Okay, here are some details about the immune systems. Observe very carefully. Okay, let's go next. There are some examples and the definitions of mast, some cells like mast cells and natural killer cells. Mast cells are the non-phagocytic leukocytes that help to initiate the inflammation by secreting histamines. In some people, histamines trigger allergic reactions as well as inflammation. Without histamine, inflammation is impossible. Mast cells may also secrete chemicals that help defending against parasites. What about the natural killer cells? These cells are in the subset of leukocytes called lymphocytes, which are produced by the lymphatic system. These cells destroy cancerous or virus infected host cells. There are some points regarding immune system. Have a note on this by having a screenshot. Okay, this is the funny example of the teeth plaques, by forming complex biofilms that provide the protection from the cells and proteins of the immune system. That means these bacteria are so clever to form protective layer so that nothing could harm them. Okay, this ability characterizes some species of bacteria and fungi. You can see an example of a bacterial biofilm on teeth in the figure below. Well, what about the introduction to innate immune system? Have a look on this, how innate immune system is boosted up and creates. There are some barriers like mechanical barriers, some first line defenses like skin, chemicals, etc. And the immune system actually are the subsets of lots of mechanical tools like the skin, the sweat glands, the fat, etc. Okay, here's the important point here. If pathogens breach protective barriers, inflammation occurs. It creates a physical barrier against the spread of infection and repairs tissue damage. Inflammation is triggered by the chemicals such as cytokinin and histamines, and it causes swelling, redness, and warmth. The complement system is a complex biochemical mechanism that helps antibodies. I have discussed detail about the complement system in the previous class. Cellular responses of the innate immune system involve the various types of leukocytes, for example, neutrophils, macrophages, and dendritic cells. Phagocytic pathogens, basophils, and mast cells release chemicals that trigger inflammation. Many pathogens have involved mechanisms that help them evade the innate immune system. For example, some pathogens form the protective capsule around themselves, and some mimic hosts cells so the immune system does not recognize them as foreign. What about the inflammation and complement system? Just have a look and have a screenshot over there. Okay, the process of inflammatory responses. There are 3 steps. At the step 1, step 1, you see the diagram 1, 2, and the next 3. Let's discuss about the 3 diagrams. At first, what happens? The leukocytes in the blood respond to the chemical attractants released by pathogens and chemical signals from nearby injured cells. The second phase is the leukocytes squeeze between the cells of the capillary wall as they follow the chemical signals to where they are most concentrated, which is called positive chemotaxis. And third one is within the damaged tissue, neutrophils release chemicals that break apart the pathogens. Monocytes differentiate into macrophages. Neutrophils and macrophages, phagocytize the pathogens and cellular debris. Here is the diagram about the innate and adaptive immunity as a comparative discussion. And this is the funny diagram of how they get activated and how they are dependent. Watch this, diagram very carefully here and have a screenshot. Okay, and let's discuss about the differences between innate and acquired immunity. what are the subjects to consider? First subject is the nature of components. Second one is how it expresses, and third one is response period. Of course, for innate immunity, the response period is for few minutes or hours, within few minutes or hours, whereas the acquired immunity after 5 to 14 days after getting the antigens and antibody. It takes a bit more time. And stability, of course, lifelong for innate, and here is from few days to lifelong. Memory responses, number 5, which is which actually doesn't happen in innate immunity and happens in acquired immunity. And 6 is the soluble components in case of innate immunity, antimicrobial peptide and proteins, whereas in acquired immunity only antibodies are the chemicals. And number 7 is cellular components. The phagocytes, killer cells and dendritic cells are there in innate immunity, whereas B and T lymphocytes work out on acquired immunity. Well, we are about to finish the class today. The homework is differences between innate and acquired immunity. But before ending the class, I must say that you must study in very details, and you have to follow more than 3 books for easily and detailed understanding. Okay, the only single video cannot work out on this, but I have tried to pick more information as possible. If anything problematic you faced, just just mention personally, and you may just share to me if you have any problems after these videos. Thank you very much.